Hello, everyone. Thank you so much
Hello.
for joining today. Hi, Harry. Great to see you. Today, we're excited to welcome Dr. Sam Dupont to be talking about Module 5 that we've been covering the last 2 weeks. He's got a great overview of so many different considerations for running biological experiments. So we're very grateful for his expertise on that front, and especially for joining at an inopportune hour all the way across the world. So thank you so much, Dr. Dupont. Would you like to introduce yourself and share a little bit about, what brought you to the OA field and what you're working on now?
Yeah, absolutely. Hello, everybody. So good day, afternoon, morning for me. So it's 1 a.m. here in Sweden. So my name is Sam Dupont, and I'm a professor at the University of Gothenburg in Sweden. And I also work half-time at the moment at the Ocean Acidification International Coordination Center in Monaco, where I work on the capacity building program down there. So my interest in oceanification is really related to biology and how we bridge between the chemistry and the biology. And over the last years now, we are really interested in the modulating role of multiple stressors and variability, and basically try to be a little bit more realistic in understanding the relationship between the chemical change, the local adaptation, and biological response. So over the years, we developed best practices and organized courses and so on. So I'm really excited to be here, and I'm looking forward to your questions.
Thank you so much. So we do have a couple of questions from the discussion board, but for anyone who's joining today, please feel free to pop any additional questions in the chat, or if you'd like, you can raise your hand and we can have more of a dialogue today. That would be great with the number of participants that we have here. But we can join or just jump right into some of the questions that our participants have had. The first one is about the longevity of an experiment. So how do you determine how long you should be running experiments? What's a reasonable time duration to ensure that the observations that we're making at the end of the experiment or throughout the experiment are valid and not just a short-term response to these conditions?
That's a great question. And actually, it's a good way to illustrate one of the problem in experimental design is that whatever experiment you're going to do, it's wrong one way or another. So that's the thing. You have to take tough decision when you design an experiment. And the answer about like how long you should perform your experiment really depend at the end of the day on your question. Ideally, what we want to do if we want to understand acidification is run experiment over decades. But because that's really what's happening, it's a slow process. Of course, it's not really possible to do that. And we should not even try to do that because we need solution within 10 years, 20 years. So we want experiments to provide answers quicker than that. So it really depends on what question you want to answer. So for example, if you're interested in multi-generation experiment, you want to know what's happening over multiple generations, you will have to do a long experiment, or you could be smart and choose an organism that have a short generation time. So you can actually do multiple generations in a reasonable amount of time. I always say, like, if you want to do multi-generation or works on the bacteria or copepod or something that you can do multiple generations shortly. But then let's say you're interested in one particular species and you want to know what will be the impact of decreased pH or increased carbon dioxide. It's important to understand how long you need to expose them to have a true acclimation to it. So for example, some of the early work that has been done on mussels for a couple of hours. That's of course more a response to a shock rather than a long-term chronic exposure. So it's recommended to do that over a few weeks, for example, to allow the species to acclimate. But we've shown, for example, on sea urchin that the full acclimation can take up to 5-6 months in certain organisms. So I don't really have the answer for you, but what you should do, and that's kind of a general rule when you study biology, is that if you don't really know, if you can't find information from the literature or from the physiology of your organism, do pilots experiments. So what you can do is like you don't start designing like a really big, really long experiment. You do it for a couple of weeks and you simplify your design, and then you take, let's say you're interested in respiration, you measure it every 2-3 days for a period of time, and you're going to see at some point when your response is saturating, when you have something stable. So it's either you talk to experts, you read in the literature, or you understand, you know your model really well. And if you don't, you do some pilots. But basically, what is really the key message here is think about your question first. I think that's a general thing for your design. If you have a good question, that's going to answer like how long you have to perform it. But because it's species or ecosystem specific, I don't know if the person who has the question is here, but you can also tell us like what species do you have in mind, and then maybe we can go into more detail.
Yeah, please feel free to chime in if anyone has any follow-up questions to that. Definitely, I've had this question about duration, before also, since running OA experiments can take a lot of energy out of you, right? With constant monitoring and keeping the animals happy, it definitely is something to keep in mind with your ability to do all of that in addition to, running the experiment well.
Absolutely. Yeah, and it's interesting what you're saying about keeping your animal happy. Like we have this big project where we want to do the same experiment in different parts of the world. And the first thing we said, okay, you have to select an organism to work on. And sure, first thing you want to be sure that you don't stress the animal in the controls that you have, because if the animal is already under a lot of stress, then you're going to see things that are not realistic. You might overestimate the impact. Or if they are too happy, which is not good either, because a lot of experiments are using, for example, really high concentration of food because it's easier to keep the animals happy with a lot of food. But then that's not something that is realistic either, and you underestimate the real impact. So that's the thing. You want to keep your organism happy, but not too happy. So that's always the fine balance here.
That's an excellent consideration too. Thank you. We had another question about your discussion of additive responses, synergistic responses. So the question was, for additive curves, we see an improved initial performance followed by the drop. Could that be a case of adaptability based on initial reaction? If you have the same species, multiple generations, and you're running it over and over and over again, perhaps, something like a bacterium or a copepod, can they finally become immune to these OA conditions and made the curve look stable eventually? What are your thoughts about interpreting in that sort of instance?
So this is a complicated question, I would say. I read it a couple of times to be sure that I understood what was the question. And I think it's mixing different concepts. So I think when we talked about additivity or things like that, it was in the context of performance curve, which is basically you measure a parameter over a range of different pH or PCO2 or whatever parameter of the carbonate system you're studying. And then you have traditionally this increase in performance till a certain tipping point, and then it's collapsing after that. So that's illustrating that over a wide range of pH, let's say pH, for example, the performance change. And then every organism, every ecosystem have a limit. So basically you hit at some point a wall. So it could be a physiological tipping point, it could be an evolutionary tipping point, and then things go bad. So you have a range where you're happy, and then if you go further than that, you go into stress and you start to have issues. The question, if I understand it well, is about like, can we explain the shape of the curve with activation or adaptation? And I don't think that's the case, because most of the time you measure it for one given parameter. So it could be respiration, could be feeding, could be whatever parameter you measure. What can happen with activation and adaptation is that the shape of this curve is changing. So if you have selection happening, what can happen is that the whole curve is shifting. So the limit of what the organism can cope with is moving, so the organism can get a little bit more tolerant. So for example, if you have here in Sweden, for most of the species we study, this tipping point is about 7.6, 7.5. So it's kind of the limit of what they can cope with. But we've done experiments showing that if you keep them over multiple generations, you're going to have some of the genotypes that are getting selected. And then the tolerance can go a little bit lower. So instead of 7.5, you're going to have 7.4, for example. So you can have a shift in the curve. So that can happen, but I don't think it will ever happen that you're going to have a completely flat curve for all the pH, because every organism, every ecosystem has a limit at some point. There is always, acidification is really related to energy and so on. And at some point, you don't have enough energy to function, and then you go into trouble. So you have a phase of plasticity, which is the part of the curve which is increasing in performance, for example, and then you have the stress phase after the tipping point. So I hope it's not too complicated, but if something isn't clear, just let me know. I can try to explain it in a different way.
Yeah, thank you. I think you gave was it a Go On webinar last year that discussed this concept as well as in the same figures. I definitely had to watch that a couple of times for it to to really sink in. It's really
that's
helpful
interesting. Yeah, because also the person mentioned additivity, which is something that people don't always understand what it means, because additivity really has, I think there's a lecture in the course talking about the modulating factor, and that's why I talk a little bit about additivity, synergism, and all these things. But these concepts are much more complicated than people think. And that was actually funny to see because I use this questionnaire during the Goan seminar and also during conferences. It's amazing to see how established scientists don't understand this concept properly. And that's why it's important to understand what additivity means. So additivity is really like this additivity at a stressor level, that you increase the level of stress if you combine temperature, for example, and pH, but not necessarily in a mathematical way at the effect side. So that's basically what I try to explain in the seminar.
Yeah, so that's great that the students in this course are getting it built in right here. I think that's really nice. We had a couple of other questions for you. And again, for those on the line today, please feel free to add some more questions in the chat or ask any follow-up questions if you would like. But we can continue on with a couple other lines of thought. One of those are, how are best practices determined? You mentioned in your lecture that the type of equipment for an experiment is dependent on your species, and to check the literature to see what's known about that. But what if there's different thoughts in the literature? How do we make a decision on what to proceed for our own experiments?
Yeah, I organized a course last week in person, which was really fun in Sweden, and we had a lot of discussion about that. Like early career scientists don't always realize that there's a lot of contradiction in the literature, as you said, and sometimes it can be a little bit shocking that some papers are incorrect or like published despite the fact that also you have mistakes and so on. But disagreeing is a really healthy aspect of science. I think it's great that people disagree. I love it when somebody challenges whatever I say and come with sometimes exactly the opposite results, because it forces you to think a little bit. I think what is important about best practices is that you want to do things right. And there are best practices in terms of how you measure the chemistry, how you manipulate the chemistry, and how to design your experiment to be sure that it's replicated. So for me, that's where you define best practices. It's like basic things that everybody should do. So if you don't do your chemistry right during your biological experiment, you won't be able to fully interpret your data or publish your results now. So that's one aspect of the best practices. For the rest, like something that are a bit more specific to your species, to your question, then it's more a question of feeling and opinion in a way. I think there is no right and wrong. Again, as I said at the beginning, all experiments are wrong. Whatever you're going to do something wrong, and that's okay. I think that's totally fine. And the strength of a field like acidification or climate change or anything is the diversity of approaches. So it's totally fine that you take a decision that is different from the others, because actually it's a strength. It's a strength to do things differently, because at the end of the day, if 10 different persons do the same experiment in a slightly different ways, but at the end, we kind of reach similar conclusions, and the difference we observe between the papers can be explained by this or that. That's great, because then at the end of the day, we have a consensus. That's how the IPCC work, is like combining the knowledge from everybody. So I think the best practice is really how you do certain things that you have to do, and that's really important. And for the rest, I would say it's okay that we don't do things in the same way, and actually we should not even try. I think that's the best. But again, if you don't know so much about how to do things yourself at the beginning, pilots experiments is the best thing ever. Like if you're not really sure, for example, let's take an example, like you don't really know what parameter of the carbonate system is important for your organism. Is it pH? Is it saturation state? Is it CO2? And you start reading paper, like we have good example with mussels, for example. People would say, oh, oysters, like it's saturation state. No, it's pH. No, it's this. No, it's that. Or it's a combination. It's the carbonate. And then you read all that and you're really confused and say, well, what shall I actually manipulate? And And in a way, you can test that. You say, okay, it seems that there is a confusion in the literature, so maybe I should invest my time in resolving this. And some people did that. They separated the effect of pH versus saturation state and showed that in certain case it was saturation state. In some other case, like for example, different life stages would respond to different parameters. So that could be also a nice way for you to identify your question. It's like, okay, I want to understand the impact on whatever, like a bivalve, and the literature is confusing. So maybe there is something to be done there to resolve this, because we really need to understand the mechanistic aspect, the why organisms respond in certain ways to pH, to saturation state, and so on, if we want to be able to project what's going to happen in the future. So I think that's really, really important to actually, read the literature, read. I always say to my students, if you want to be a scientist, you have to read. And it's really important to read broadly. I think I read papers for work, I read books on the side. I think it's opening my mind to certain things. And I used to read, for example, Nature, at least the first part of Nature every week, even if it was completely unrelated to my field. It's going to open your mind. So I would say that could be, if you don't really know and it's confusing, that might be a good idea to define questions linked to this.
I agree. And diving into supplementary material as well. You know, I think the OA field has been really good about documenting very specific steps in the supplementary material. You know, if it's not described in the paper, and that's a good way to see some of the finer points.
I see that I had a nice comment in the chat. So thank you so much for your nice words. It's really nice to hear that you enjoyed the lecture, and feel free, by the way, if you to also send keep interacting on the chat, on the website, and so on. And you can even contact me later if you have questions or comments like specific. And don't hesitate to ask specific questions too now if you have like already an idea or something you want to do, and you have specific questions. That could be a nice way to illustrate certain points. So if you have questions, please shoot.
That's an excellent point. Yes, we are so grateful for your time joining us today. This is a wonderful opportunity to ask questions of Dr. Dupont if you're thinking about running experiments. What I would have given to be able to chat with you when I was designing my PhD work. So this is fantastic. Thanks again. So yes, feel free to raise your hand if you'd like, or pop those in the chat. But while you're thinking of those, we can talk about some other questions. One of those is statistics. You know, after we design our experiments, implement them very well, one of the later steps is then figuring out what it all means. So do you have recommendations for increasing statistical knowledge? As you mentioned, you know, sometimes we want to do power calculations or other sorts of statistics before we begin.
Yeah, there are I would say 2 points linked to this. I think it's important to think about how the general kind of strategy you're going to have for your statistics. So if you do experiments, you want to be sure that you're going to be able to analyze them afterwards, and also be sure that you're going to have something, it would be the power to actually see something significant. So on one side, I mentioned that during the lectures, you have these kind of 2 main strategies that you can use. One is kind of an ANOVA type of design where you test scenarios, and then you need to have a certain amount of replicates to be sure that you have enough power to see difference between your scenarios. So, or the other approach is to use this more regression approach where you have a range of different pHs, and basically choosing one over the other will depend once on your question, but also on practical limitations, like how many tanks do you have? I think it could be as simple as that. For example, we had a huge experiment in front of the station where I worked with mesocosms. And basically there are 10 mesocosms, that's it. You can't have more. So in that case, the best approach was to have 2 scenarios and 5 replicates, and that was powerful enough. But then if you have unlimited time, unlimited money, you can do like wide range and use beautiful regression, and both approach have their value. But then like if you want to know also like how many replicates you want to include, and because statistically, if you want to see an effect, you always have this variability between your replicates, and the more variability you have, the more replicates you need to have something that is statistically significant. So in that case, again, if you have an idea of how much variability you're going to have, then you can calculate. Actually, you have something called power analysis, how many replicates you need. So that's something you can calculate. If you have no idea, you can check in the literature or ask experts. They say, I want to measure respiration rates. And these, for example, are well known to be noisy because of you create noise while measuring. So it's technical variability. So in that case, you know you're going to need a lot of replicates. But if you don't know, again, make some tests. You can spend a couple of hours, couple of days collecting data and see how much variability you have. And then you can calculate how many replicates, and then you will know that you're going to have good statistical power. So that would, again, I know I'm repeating myself already for the last 20 minutes, but do pilots, do some tests. If you don't have the experience with the thing, do test it. And also a good recommendation that you can have is if you do something new, it's going to take you a lot of time to develop the skills. And my way of working is I try to work with people who have expertise in the field already. I wanted to measure larval swimming, for example, and had no expertise. And I thought naively that you just film an aquarium and then track the larvae, but it's much more complicated than that. And so I invited a colleague that I've met in a conference to come to work with me for months. And we did so much because we could immediately start. She knew how to set up the aquarium. She knew how to do that. And so I saved a lot of time. So it's great if you want to do a lot of things by yourself. I think it's also really cool, but collaborate. I think that would be like a good message from this lecture. Most of the cool thing I've done came from meeting people in a conference or guests here at the station where I work and really try to capitalize on our strength and work together. So if you have the opportunity within this course of creating bridge between you and say, okay, I'm really good at metabolism, I'm really good at this, I have a great aquarium system, then try to work together. You're going to save time and do much better work.
Yeah, this sort of calls back. We had a live session with some members of PI-TOA and the Pacific Islands Oceanification Center a couple weeks ago, talking about networks for, making these sorts of connections, whether that's through PI-TOA or the OA Info Exchange or the GOA on peer-to-peer network. So there's lots of resources that this community has been really, forthcoming with to start to develop those connections and find areas of expertise to be able to do more together more quickly.
Absolutely, and the ocean acidification community is very friendly. It's an easy community. People are really willing to take their time. You're going to see like people with big names like Andrew Dixon, people like that are actually super approachable. You can just contact them; they will take the time to answer. So that's great. I really like this community.
I agree. It's a great community to be a part of. Another question that we have is, how much does it take to set up and run an experiment in terms of cost? If we're putting together a budget for an experiment, what should we build into the grant to consider? And there's so many different parts in an experiment.
Again, that's a difficult question to answer. As you can imagine, I can tell you a story about this. So one of my first capacity building training I've done was in Cape Town in South Africa. I was down there with some colleagues, including Andrew Dixon, and Andrew was basically presenting the best practices for carbonate chemistry and explaining how complicated it can be to measure alkalinity, even pH. As you know by now, it's not an easy thing to measure. And the second day I was having breakfast, and one of the participants came to me and said, "Oh, that was really great yesterday." I said, "Oh, that's cool, thanks." And we'd say, "Oh, but it's completely useless." And I was like, "What?" And basically explained that all the techniques that were presented were completely impossible for them to implement because of cost and lack of infrastructure. And that's where I realized, and the Ocean Foundation also did a lot of that. We need to find another way of working based more on what people have. So now the approach we have is when we organize training is really we try to see what are the needs, what are. Sorry, there's somebody with the mic on. So
Laboratory is advanced.
Mutes. All right, I muted. So basically, we have to work with what people have. So in that case, you have to first see, okay, what kind of lab do you have? What kind of equipment do you have? What kind of budget do you have? Also, which is a very important question. But in that case, what we came up with is that you can do things with very little. So let's say that you don't have a lot of budget to start with, or you can only have access to a small grant. That's fine. You can still do excellent science with very little. So we developed, for example, some fertilization assays that can just be done with like plates, plastic plates or small jars, and we do that on the east coast of Africa, and that's really successful. I think you can really, so basically you can, with a couple of €1,000, I would say, you can do something. What you really need is a pH meter minimum, I would say. Then, as you know, it's better to calibrate with tris buffer, but there are methods to do it yourself. So that's not too hard. Titration you can do with manual titration. So if you have a pH meter, you can and a minimal chemistry lab, you can do your chemistry. And then for the manipulation of the water, it can be done in a very simplistic way, just mixing water and so on. And I think I was mentioning in the lectures, like all the different techniques. So you can do it with a really small budget, and then you can go crazy on the other side. Like we had this mesocosm experiment that I just mentioned, that we are talking about $1 million or something like that to run such an experiment, because it's 50 people working full time. It's massive infrastructure. So that's the two sides of it. But I would say, let's say if you start from scratch and you want to buy a couple of pH stat systems to have, let's say, about 8 tanks, which would be nice. You don't really have a pH meter. I guess a budget of $10,000-$20,000 would be a good start, something like that. But again, that you can do with less, you can do with much more. I started with 2 pH stat systems, so I had 2 replicates at low pH and 2 controls. I think it would be hard to sell that today, but that's basically what I had when I started, and I published my first paper with that. So it was €1,000 budget or something like that.
Yeah, for my own experiments, I used like a home aquarium system. I think that was around $1,000 or so. It worked really well enough. But I think the nice thing about OA experiments too is that sometimes measuring the responses can be relatively inexpensive, right? If you're just measuring survival or growth, you don't necessarily have to do very expensive like bioinformatics. You can if you're interested in that, but there's definitely a range on the responses measured to that different associated.
Actually, it's fun that you say that, because again, I keeping coming with the same message, but the question is more important than the technique. So you can actually answer really cool question with, as you said, very simple endpoints like fertilization, survival, growth, which are very important. And then you can see sometimes some papers where they invested a lot of money doing like a whole transcriptomic and or proteomic, without a very serious question. And at the end of the day, the added value of adding these parameters is very limited. So it's a lot of money, a lot of time, a lot of samples, a lot of everything. But if it's not rooted in a really key question, that's not necessary. I think that's a bit provocative to say that, but there are a lot of data out there that I don't think contribute too much to the field. And it's more because people get excited about techniques more than about really questions. So think about, don't I would say to the group here, I don't know what kind of facilities you have, I don't know what kind of budget you have, but if you can publish really cool and interesting data if you really focus on the right question. And there are tons of questions to answer, especially where you live, where the local data are quite limited. So there are a lot of really cool stuff that needs to be done really, and that you could contribute to with whatever you have.
Yeah, thank you so much for sharing more about that. One thing that I'm curious to hear is, where do you think the field will be in 5 or 10 years, right? You have a wealth of experience that you've seen the OA field develop over the last couple of years or so. But where do you think we will be in the future? Will we still be doing single species experiments? Do you think it's more valuable to shift towards like mesocosms or more ecological based work? Or is there still value in filling some of these these like gaps in single species?
Yeah, that's interesting because I think you touch on two things. I think they are the question again. So I think from a question point of view, they are the field is evolving in like these three directions that you mentioned. So first, people complexify. So you move from one stressor to multiple stressor, and that's going to be a big question for the next 5 years to answer because it's extremely complex. So I think what we move definitely, and you can see that already, there are more papers combining multiple stressors or multiple drivers. Definitely also going from this single species to community to ecology is also something very important to answer, and also increasing the time as the third factor, like including like evolution and also variability into the questioning. So that's, I would say, like that's philosophical in a way. Like it's like what kind of questions we can answer. That doesn't mean that we need necessarily to move from experiment on single species to mesocosms, for example. You can answer ecological question in an aquarium without necessarily have to go in the field and make complicated things. Or you can answer complex evolutionary question in a simple setting, or you can still, single species experiment are still valid for many questions, including multiple stressors. If you don't understand how an organism responds to one single driver, you won't be able to understand how they work with multiple. So there, I think it's not more a question. I think the field will evolve to more complex questions, but not necessarily more complex experimental settings. So there's, for me, all the different approaches are complementary. So we will need single species lab experiment. We're going to need mesocosms. We're going to need field. We're going to need monitoring. All these things are still important. So don't be scared also by the fact that, oh, I can't do a mesocosm. I can't do mesocosm by myself. It's too complicated and too costly, so I don't do it. So even if you can do simple species experiment over a short period of time, that's still interesting, and that's a complementary approach. So again, the answer will come from the diversity, from working together in collaboration, the diversity of approach, the diversity of endpoints that you're going to measure, the diversity of questioning. But that's basically where the field is moving to. I would say personally, what I hope is that the field is going to move into something a little bit more applied, in the sense that I'm not the one to judge if you decide to do pure basic science or applied science. I think that's something that both are really valuable for sure. But acidification is a societal issue. It's going to have consequences for us. It's going to have consequences for many people around the world if we don't address it. So we should prioritize, because we don't have time. We don't have 100 years to fix this. We need to have data within 10, 20 years. And we need to be able to provide recommendations to policymakers, to industry, to basically all the people using the ocean. And to do that, some specific science will be needed. So it's not just about doing whatever we want just out of curiosity. That's valuable, of course. But because we want really to provide options and solutions to address the issue, we need to prioritize. And that's what I hope, that the field will be more and more moving into prioritizing the science that can have an impact. And for example, I can see more and more people at the moment studying alkalinization, which is the idea of, increasing the carbon, the alkalinity of the water through different ways with olivine or other minerals. That's one way to potentially increase the carbon capture in the ocean, also maybe make some ecosystem more resilient. I think that, I'm not sure if it's working, but that's worth testing. And I think that's also an interesting question. But if I say, like, personally how I do it, I actually cheat a little bit. So I do a little bit of both. So I try to do science that is useful, that in the context of acidification, but also in the same time try to answer classic biological questions. So for example, we were talking on local adaptation. That's a classic concept, or the cost of variability. So you can basically play a little bit with both. And also you have to be smart. I don't know where you guys are in your career, but you have to be strategic at the beginning of your career to be sure that you're going to get a job. So you have to be smart in that way. And later, even if you have a permanent job like I do, you still need to get grants, and you have to sense a little bit what people want. And at the end of the day, I try to play the system in a good way. So I try to deliver what is needed, but at the same time do things that I believe is important. So that's, if the grants are more about basic science, you do something about basic science, but within the back of your head doing something useful. And if it's the other way around, you can do that too.
Great. That point about prioritization is so important. I'm curious to hear a little bit more how you as a scientist, how you've kept your ear to the ground with policymakers or stakeholders that are perhaps interested about changing ocean conditions, and learned where those sort of avenues are that you could perhaps contribute your research towards learning more about some of these issues that folks outside the scientific sphere are interested in.
I can talk a little bit about my personal experience along those lines, but I think the trick is to reverse a little bit the way we do science. So we tend to do the science that we think is important by reading the scientific literature, talking to colleagues, and so on, which is fine, of course. But the thing is that most of the time we do that, we produce science, we publish paper, and we communicate that to the world, like policymakers, industries, and so on. I think it's much more efficient to do it the other way around. So you basically start by talking with the people who will use your science. So you go and you talk to policymaker and say, okay, what kind of data do you need to actually implement regulation or management? Or you can go to a local aquaculture and say, okay, what problems do you have? What options do you have? Or you can, I think there are a bunch of people using the ocean, and talking to them first will give you an idea of what is really important locally. I used to work a lot in Hong Kong, where they have this oyster farmer there, and that all their livelihood depends on one resource, which is this local species of oyster. And what we did is my colleague went there and talked to them. So, okay, what are the issues? And then really they didn't like scientists, these guys, because scientists are the people telling them you can't sell your oyster because it's polluted, because it's too toxic. So we went there and said, okay, we are not here to do that. We are here to listen to you and try to see what the problems are and what we can do to help. And then we realized at the time there that acidification was not the main issue today, but it was heavy metal pollution. So we say, okay, let's try to find a way to detoxify the mussel, because we can't really solve the problem in the ocean because it's polluted and it's going to be polluted. So we say, okay, can we move the production on land? And so that's, I would say that would be my trick. Go and talk to people, see what is needed to, and what kind of science is missing to implement solutions over a reasonable amount of time. So not in 100 years from now, but in 5 years, in 10 years. What do I need to understand to manage the ecosystem, to protect the resources, to keep the industry going, and so on? I think for me, that's been really interesting. And for example, if your goal is, I want to convince the public, that's also a very valuable way of identifying a cool question. So I want to, on my island, I really want to influence the people in the society around me. Then you also have to go and talk to them. We did that in Sweden by interviewing people in the street and asking, okay, what do you care about to the ocean? And they said, we don't like jellyfish. So they hate jellyfish here in Sweden because they bloom during the summer and it's really unpleasant if you go swimming. So that was one thing. And the other thing, of course, it's seafood. They really like the shrimps here, the lobsters. And so we decided, okay, if we want to convince these people that something needs to be done in terms of climate, in the carbon emission, and how the ocean is managed, we need to speak their language and basically work on things they care about. So that's why we did studies on shrimps. That's what we did studies on jellyfish, is really with this idea that this information will be more efficient at driving change at different levels.
That's great. We're chatting before we started today, and you mentioned there's a communication course that you're looking forward to putting on in the future. What are some avenues that you found, once you've heard from those in your community what they're interested about and done the experiments, what are sort of the ways that you found to share this sort of information back to those folks who are interested?
Yeah, that's a difficult part of the job, isn't it? I think when you're a biologist or a chemist and then you found something that you think people should care about and then communicate that to an audience, that you're not used to is very hard, and it takes years of practice. I would say for me, I've never been trained for at the beginning, so I had to train myself. I took courses and so on. But the thing is that every time you talk to a different audience, it's a different language, it's a different expectation. So that's what I like about it. I do more and more of this because every time you have to reinvent yourself in a way, and you have to learn the language. And this course we're going to organize hopefully in October in Costa Rica, and I think you guys keep an eye on the OAICC news stream. It's going to be advertised there when it's ready. The idea is really to do some practical, so to put people in the room in presence of professionals, like could be journalists, could be policymakers, could be people in the industry, and learn really the language first, and then develop some efficient ways of communicating the same message to different audience, because people don't have the same expectations. Like, don't go with a problem to a policymaker, for example. They don't want problem, they want solutions. So you have to come with a twist and say, okay, acidification is a problem, but this is what you can do. And be really clear about that. If you go to people in the street, you need to trigger emotion response. I think if you just go with your message like my lobster respiration rate decreased by 50%, nobody cares. So you have to turn that into something that people will understand and care about. And if you talk to a journalist, come with a really cool story. I think that's an easy one, but you have to come with a story that they will be excited about. So yeah, it's keep an eye on the OAICC, so Ocean Acidification International Coordination Center news stream, and as soon as we have a concept ready, we will post an announcement, and it's going to be open for everyone.
Perfect, thank you. We'll share a link to the news stream in the follow-up to this. I believe it's in the resources as well that was shared after the live session for resources for oceanification. So it's really great. Thank you for letting me take advantage of your far-ranging knowledge in the OA field. I do want to bring it back just a little bit more technical to one of the quiz questions. So for those of you who haven't completed the quiz yet, this is your bonus for attending today. In terms of how you sort of create OA-like conditions in the lab, what are sort of the different avenues that you've done or your colleagues have done to replicate, this process that's going on in the oceans over a long period of time in the lab in a short period of time, whether that's adding gas or other reagents? What can you share in terms of these different avenues to do that?
Yeah, sure. I think the two main ways of doing that is adding carbon dioxide, which is basically what's happening out there, right? You have more carbon dioxide in the atmosphere, so that's increasing in the water, or play with carbonate and acid, basically, by carbon. So the CO2 is relatively easy, and what you can do is either manipulate the concentration of carbon dioxide or controlling the pH. So I think there's a lecture where I explained a little bit that in more details, but you can have pH stats that measure the pH and bubble carbon dioxide when needed, and you can actually keep the pH relatively stable with that. We set up an experiment like that today, and we could keep the pH stable with a 0.02 pH unit stability. So it's a cheap system. It's just a few €100 for. And you can do that. So the other way is you can have a mixed gas where you basically bubble in your system with gas with a known concentration of CO2. So it's keeping the CO2 constant. And then there are multiple ways you can use these tools to, for example, you can also create variability. I think that there are multiple ways of doing that. But then the other way is, let's say you don't have any of those, but you have a good chemistry lab. Well, what you can do is also manipulate a bunch, like you can have a bucket full of seawater, and then you can add strong acid, like HCl, for example. But if you do that alone, just HCl, the problem is that it's not really replicating what's happening with carbon dioxide. You're also changing the alkalinity. So if you just use CO2, it's keeping the alkalinity constant. If you add strong acid, the alkalinity goes down. So in that case, you need to correct the alkalinity by adding carbonate, for example, play with carbonate and bicarbonate. So that's that's the cheapest way of doing it if you don't have any equipment. It's a little bit tedious because you really have to be careful about how much you add, but you can also do it with trials and error, and it works it works kind of okay. Something you should not do for sure is just add acid because you change the alkalinity, or just use carbonate and bicarbonate, because if you do that, you're going to basically also manipulate the pH in the wrong way by increasing the alkalinity. So acid decrease the alkalinity, carbonate, bicarbonate increase it. So if you do use both, it's fine, you can compensate. But if you use one of it, it's wrong. And the problem with that is then you simulate something that is not exactly what acidification would be. So you have confounding factors, and you're going to see something for sure, but you won't be able to know if it's really simulating acidification and something that will happen in the future, or can most of the time would be over. You're going to have an over negative effect compared to what you want. So there are papers where they compared both, and they could see that it's different. So if you just use acid or CO2, you don't have the same results, and acid is stronger negative response. And to be honest, if you try to do that, you won't be able to publish these days, because nobody, I think it's going to be spotted immediately as not the best practice. So it's really important not to do that. So that's a good point that you mentioned this. Don't do just acid. Don't do carbonate bicarbonate only, because that doesn't work.
Great, thank you so much for explaining more about those different ways to create these sorts of conditions. We're coming up a little bit to the end of our hour, but we still have a couple of minutes left. If you have any questions, feel free to add those in the chat, or raise your hand. This is a space where any sort of questions are welcome. We have a lot of different participants in the course ranging with different backgrounds, so please don't be afraid to ask anything that you think others might know, or we're all learning different things here. I'm still learning a lot just from watching the lectures each week myself. So you can wait for any of those to come in, but I'll just note as a reminder, the quiz for Module 5, Dr. Dupont's lectures due this Sunday, so make sure to complete that. Next week, we'll be learning about chemical observations in the field with Dr. Adrian Sutton, who you might remember from a previous module. And then the week after that, we'll be finishing up the course learning more about biological observations in the field. And Dr. Dupont will be returning again late at night for him for another Q&A with Dr. Finley as well.
And many of the things that you have in the biological module here will be very useful for the biological monitoring part, because we basically apply some of these concepts there to, for example, you can estimate like how long you need to do your monitoring if you hope to see an effect. And that's based on concept that we presented in this module. So they're going to be really close link between these two. If you want also to share maybe your experiences, or if you had technical issues, if you tried an experiment, we can also talk about that. And again, don't be shy. I'm more than happy also to continue the conversation offline after. If you have questions, you can put it on the forum, or you can drop me a message, and we can take it from there. And if I don't answer, if ever one of you write me an email and I'm not answering, don't be offended. Just send me a reminder. I tend to be terrible with emails. I have too many of those, like everybody, I guess.
Yes, for sure. Yeah, feel free to add those in the discussion forum. We'll make sure to send them over to Dr. Dupont. That would be really wonderful. Thank you so much for, first off, giving the wonderful lectures for this module. It's been, such a great amount of information that I think we'll all be able to return to over and over again, especially as some of us begin running biological experiments for the first time. Great to return to this resource and find those little details that perhaps we didn't catch on the first watch or so. And again, thank you so much for joining us today.
My pleasure. I think I'm going to go to bed now. It's going to be 2 a.m. soon. All right.
Thank you. Have a good
Thank you very
day,
much, everybody. Thanks for joining.